who just joined, our learning outcome is, the first one is the definition of environmental microorganism. The second one is the roles of microorganism in environment, and the last one is classification of bacteria. So, and we have just defined the term environmental microorganism. These are microscopic organisms that includes bacteria, fungi, protists, archaeas, and viruses that inhabit various natural and artificial environments. So it simply means microscopic organism means they are very small organisms that we cannot see in our naked eye. So we use microscope to be able to see them. Let's look at the roles of microorganism in environment. The first role is decomposition. So this is whereby bacteria break down organic matter such as dead plants, animals, and waste materials. The second one, they aid in nutrient cycling. How? They convert complex organic compounds into simpler forms that can be used by plants and other organisms. The third one, they help in bioremediation. That is, they have ability to degrade or detoxify pollutants in the environment through processes like biodegradation. And then we have the other one, they help in waste treatment. Irene, can you read for us how they help in waste treatment?
Waste treatment to break down organic matters and remove pollutants from water.
Thank you very much. And Nicholas, can you read for us how bacteria aid in biocontrol?
Okay, biocontrol uses biological control agent to manage pests and invasive species in agriculture and natural ecosystem.
Very good. Thank you. Can you clap for Nicholas? So simply, they bacteria used as biological control agents to manage pest and invasive species in agriculture. Instead of using pesticides or herbicides, we use this microorganism. Then let's look at the classification of bacteria. First of all, bacteria are classified according to 4 types. The first one, they are classified according to the shape. We also have a classification of bacteria according to the cell structure. We also have classification of bacteria according to metabolism, or we simply call it nutritional needs, and also classification according to environmental preferences. We also have classification according to special features, and classification according to oxygen requirements. Let's look at the first one, classification according to shapes. So you realize that bacteria are in form of different shapes. The first one, cocci shape. These are spherical bacteria, and they include staphylococcus. We also have bacilli. These are rod-shaped bacteria, for example, Escherichia coli. As I explained, I hope you are able to identify in these pictures which one is a cocci shape, which one is a bacillus shape, which one is a vibrios and spirilla, and so forth. So you should be listening as well as observing what is in the pictures. So the third one, we have spirilla shape. These are spiral in shape. These are bacteria which are spiral in shape. And you can see here, this is the spirilla. They are spiral, or they are rod shaped like. Then we have another one, vibrios, that is comma shaped bacteria. And an example is Vibrio cholerae. And this Vibrio cholerae is responsible for causing a disease called cholera. I'm sure most of you are familiar with the cholera, especially those who uses if you drink dirty water, you are likely to be infected with cholera, and that is those dirty water contain Vibrio cholerae, and that's why you realize you will be infected with cholera. We have another shape, the last one, which is filamentous shape. Filamentous shape, these are thread-like bacteria, and an example is actinomyces. This is an example of the bacteria which has a thread-like shape. Let's look at the classification of bacteria according to cell structure. So let me see these people who are present in class. We have okay, I can see someone called Nelson. Nelson, can you read for us the classification of bacteria according to cell structure? Unmute your microphone, please.
Thank you so much. So I'm saying gram negative and gram positive. And for gram positive, I take peptidoglycan cell wall, e.g. this Staphylococcus aureus. And the gram negative, thin peptidoglycan layer with outer membrane, i.e. the Escherichia coli.
Thank you very much, clap for Nelson. So we are simply saying we have gram positive and gram negative. For gram positive bacteria, they are thick. Sorry, can you mute your microphone in this one? Please mute Nelson. Please mute your microphone. Yes, thank you very much. So we are saying gram positive. They are thick peptidoglycan cell wall. That is, they have cell wall which is thick. And also an example is Staphylococcus aureus. And then gram negative, they are thin. So the difference is the gram positive is thick wall and gram negative is thin wall. So let's look at the other classification, which is a classification according to metabolism nutrition, according to metabolism or simply nutritional needs. So we have autotrophs and we have heterotrophs. Autotrophs, they produce their own food. An example is cyanobacteria. And then heterotrophs, they obtain nutrients from organic compounds produced by other organisms. I hope you'll be able to differentiate autotrophs and heterotrophs. Then we have classification of bacteria according to environmental preferences. And this simply we have psychrophiles, we have thermophiles, and halophytes. Can someone tell me what does it mean when we say thermo and halo? These are the terms you are familiar with. Yes? Kibi what?
Thermophiles.
Okay, yes, Faith.
We have thermophiles which prefer high temperatures, kama thermos.
Yes. So thermophiles, it means thermo means high temperatures. So thermophiles, they grow or they survive in high temperatures. And then halophiles, that is salt-loving bacteria. They survive in a salty environment. And then psychrophiles, they thrive in cold temperatures. An example is Psychrobacter. Kibiwat, can you read for us the last one, which is according to special features? Isaac Kibiwat, are you in?
I'm sorry, I'm not able to see your screen, madam.
Class, can you see my screen? Who else is not able to see my screen?
We can see.
It's visible.
Okay, so Kibiwat, it seems you're facing some problems with network, maybe. Because if the rest are able to see, then it means maybe you have the problem on your the problem is on your side. Can you raise up your hand? Maria Viane, are you able to read for us? Can you read for us?
I can now read. I've now seen it.
Okay, thank you, Isaac. Read.
Classification of bacteria, is it?
Yes, according to special features.
Yes, according to special features. The first one is spore forming, whereby it's the ability to form spores to survive harsh conditions. These examples are Bacillus and Clostridium. The other one is motility, which is the presence of flagella or other structures for movement. Examples are Escherichia coli and Treponema pallidum. And then we go to pathogenicity. Pathogenicity, ability to cause a disease in host. Examples are Mycobacterium tuberculosis, and then Staphylococcus aureus.
Thank you very
And the
much,
other one,
Kibiwa. Can you clap for him? Yes, you have another one?
is the classification of bacteria according to oxygen requirements.
Yes.
We have the aerobes, which requires oxygen for growth. Examples are the Mycobacterium tuberculosis. And then we go to anaerobes, which do not require oxygen and may be killed by it. Examples are the Clostridium. And the last one is the facultative bacteria, which can grow with or without the oxygen. An example of it is the Escherichia coli.
Escherichia coli. Thank you very much, Kibiwat. Can you clap for him? So we are simply saying these are bacteria which are classified according to oxygen requirement. There are those bacteria which require oxygen to survive. Without oxygen, they die. And an example is Mycobacterium tuberculosis. This is a bacteria which is responsible for causing tuberculosis disease. That is an example of aerobes. Then we have anaerobes. These are bacteria which do not require oxygen, and if you subject oxygen to these bacteria, they die. And then we have those bacteria which survive whether you subject to oxygen or not, they still survive. Those are facultative bacteria. An example is Escherichia coli. So this marks the end of our class today, and I'll be taking the roll call. I expected that everyone will be in by the end, so don't leave the class until I dismiss you officially. So thank you very much. Now let's summarize. What have you learned in our session today? What have you learned? Yes, Faith?
We've learned about the classification of bacteria by cell shape, metabolism, nutrition, and environmental preferences.
Thank you. Clap for Faith. Yes, Nicholas, what have you learned today?
Classification of bacteria
Uh,
according to the
we have also learned about the classification of bacteria based on the oxygen requirement. That is facultative ones and the
Aerobes and anaerobes. Very good. Thank you, Nicholas. And then Kibiwat, you want to say something? Isaac, do you want to say something? Oh, he left already. So thank you so much for your participation. I appreciate. So I've taken the register. I'm giving you an assignment whereby this assignment is 20 marks, and you are supposed to return in our next class next week. It's part of our Cat 1 assessment. So the assignment is classify characteristics of protozoa and fungi. 20 marks. Classify characteristics of protozoa and fungi. So thank you so much, guys. Bye. See you next week. Let's send appreciation. Send the flowers and love emojis.